My name is Michael Reich, and I direct software development for the lab of Jill Messerov at UCSD. I'm going to describe the features of the GenePattern notebook environment, what they provide above and beyond the standard notebook experience, and demonstrate a number of notebooks we've created for genomic analysis that can be viewed, run, and adapted by the scientific community on our online notebook workspace. By now, everyone in any field related to data science is familiar with analysis notebooks. Although notebook-based systems have existed for many years, the Jupyter Notebook environment was the first to capture the wider needs of data scientists and scientific users. This is due to the unique benefits of the notebook environment of reproducibility and support for both live code and associated text and images within a single document. However, for genomic and bioinformatic analysis, the notebook metaphor presents a number of challenges. The first is that notebooks assume that the user is a programmer. To use and understand notebooks, a scientist needs to have a background in a language such as Python or R, and familiarity with the packages that provide analysis and graphing functionality. Many biologists and bench researchers who could benefit from the features of notebooks are limited by their programming experience. This is especially true in cases where bioinformaticians share notebooks that are useful but require the user to modify the code to adapt it to their requirements, even if that's only to change the parameters of a function. And even the entry of formatted text is done following the programming paradigm of the markup language to express formatted text. For example, in Markdown, to create a table that looks like this, you need to write text that looks like this. And in LaTeX, to create mathematically formatted text like this, you need to write code that looks like this. There's also the challenge of providing access to existing bioinformatics methods. Many of these are command line tools, so you could install them into your Jupyter environment with all of their dependencies and maintain them that way. But that prevents your notebooks from being adopted by others who may be on a different platform. Also important is that many bioinformatics analyses are compute intensive. You might want to create a workflow that includes RNA-seq analysis, non-negative matrix factorization, or other tasks that might overwhelm the compute capacity of the machine running your notebook server. This is especially true in environments that provide hosting for multiple notebook users. It would be ideal to offload compute-intensive analyses to the resources best suited to support them. To summarize, we need easy access to existing analysis tools in a way that doesn't require ongoing maintenance of each tool. An easy way for non-programming scientists to access the methods in notebooks developed by bioinformaticians. Tools that don't require knowledge of a markup language to create richly formatted text. The easy publication and dissemination of notebooks, because it would be ideal for other users not to have to download and run a notebook locally or use Binder or a similar service that limits a user's ability to run and modify a notebook. And finally, for common workflows, users shouldn't have to start with a blank page. There should be templates supporting common bioinformatics analysis scenarios that scientists can easily adapt to their own objectives. These are the requirements that led to our development of the GenePattern notebook environment. This environment gives you all of the notebook features of Jupyter and is fully compatible with Jupyter and not a separate fork of the code. The GenePattern notebook environment adds the ability to utilize analyses from any GenePattern server, in addition to a number of other general features to make it easier both for non-programmers to have access to the full range of genomic analyses required for today's integrative genomics experiments, and for the notebook developers to make it easier for their own notebooks to be more accessible to users at all levels of computational sophistication. GenePattern wraps analysis methods as accessible web forms, providing all the features of the analysis without the need to know the programmatic implementation or to provision the necessary compute resources. The main GenePattern server runs on the Amazon cloud, utilizing the elastic compute capabilities of AWS to run analyses on the most suitable resources, for example, compute nodes with high memory or rapid data transfer as required. The main GenePattern server hosts hundreds of analyses, which we add to frequently based on our scientific collaborations and user requests, providing a considerable cross-section of platform-specific methods as well as general machine learning approaches. There are also other dedicated GenePattern servers that provide access to high-performance compute resources to run especially resource-intensive algorithms. You can connect to any number of GenePattern servers from within a GenePattern notebook, combining the unique strengths of all of the publicly available resources in the GenePattern ecosystem. Let's look at the features we've added to the Jupyter Notebook system to enable this functionality. A GenePattern login cell allows users to access any available GenePattern server. That means the public server on the Amazon cloud or the UCSD Expanse supercompute cluster, for example. Or if you run your own GenePattern server, you can connect to that as well. When you want to find a GenePattern analysis to insert into the notebook, you create a new GenePattern cell in the same way you would create a code or markdown cell. You can use the menu navigation or keyboard shortcuts. Once you select the GenePattern cell type, you'll see a browser that allows you to choose the analysis you want to insert. You can either scroll through the list of available analyses, or type an analysis name, or type to filter the list of choices, for example, clustering. Once you've found the analysis you want, click to insert it as a cell in the notebook. Once you fill in the required inputs and run the analysis, you see a panel showing the status of the job, and when it's done, it presents you with the result files, which you can easily use in downstream analyses. As part of our seamless integration with Jupyter, you can use a Python variable in any suitable input parameter by enclosing it in double braces. Once you launch a job, you will see a job cell that displays the status of the job and gives you access to result files. A job always begins in the pending state. When it's launched on the compute cluster, it enters the running state. When a job is finished, it enters the completed state and displays any result files that were generated. Clicking on a result file will display a number of actions you can perform on that file. In addition to downloading or viewing the file, you can load its contents to a Python variable or a Pandas data frame where appropriate. Or you can send the result file directly to another GenePattern cell in the notebook, or view a list of analyses that can accept that file as input and create a new cell with the result file as its input. To further increase ease of use by non-programmers, we've developed a user-friendly interface to Python functions. So that notebook developers can provide a clean interface to their functions that expose only the parameters that are required to run them, we've developed what we call the UI Builder. This allows notebook developers to add one decorator to their functions, which describes the inputs that they want the user to be aware of, and to then automatically create a web-based interface for that cell. This hides the complexity of the code and makes the functionality much clearer. Developers can specify file inputs, drop-down lists, and can tailor the interface in many ways to the specifics of their method. You can click a button to toggle back and forth between the code and UI Builder views, and any changes to the code are automatically updated in the UI view. You saw earlier that even just creating text in a notebook requires a programming type perspective. We addressed this as well. We have added a rich text editor that lets you add text as you would any word processor. This is an added feature and doesn't replace the other ways of adding text. This feature makes it easy to add elements that are common in scientific literature, but difficult or impossible using the native Markdown language supported by Jupyter, such as superscripts, subscripts, and special characters. You can easily toggle between rich text editor, HTML, and the final rendered text view. Scientists can create, run, and collaborate on notebooks using the GenePattern workspace, notebook.genepattern.org. This is a free resource where you can work with notebooks using only a web browser. The Notebook workspace features a library of notebooks that scientists can copy into their accounts and run as ready-made analysis templates, or edit to their requirements. Available notebooks include genomics analysis workflows in gene expression analysis, single-cell RNA-seq, multi-omics analysis methods, gene set enrichment analysis, general machine learning methods, as well as tutorial notebooks that teach the functions of the environment. In addition to our featured notebooks, there's also a section containing notebooks contributed by the community of GenePattern notebook users. When you make one of your notebooks public, It's available to other users via the Community tab. Let's look at a few of the notebooks that are available in the public library. Due to the popularity of single-cell RNA-seq analysis, we'll highlight some of the available notebooks that support several different scRNA-seq approaches. A number of single-cell software tools provide analysis workflows in which users examine intermediate results at each step as part of an interactive process. The developers of the Seurat toolkit in the Raul Satija lab authored a tutorial walking users through downloading, pre-processing, clustering, and performing differential expression on a dataset of peripheral blood mononuclear cells. We adapted this workflow to the GenePattern notebook environment and made it available for those that want to run the Seurat pipeline on their own data. The notebook section being demonstrated allows users to upload single-cell data in any of the commonly available formats and displays a visual representation of various common quality control metrics used for filtering single-cell data. We have also implemented a pseudotime method called Stream from the lab of Luca Pinello using GenePattern modules and notebooks. Pseudotime analyses use techniques such as dimensionality reduction and applications of graph theory to identify these processes in the data and order the cells along a pseudotime trajectory. As a note, in this ordering, the distance between one cell and another does not indicate a difference in terms of real units of time. It only indicates which cell comes after another in the sequence of a specific biological process. On the left, you can see an example of Stream's intuitive pseudotime representations, which the authors call subway plots. In these plots, you can see both the different branches of the trajectory as well as the density of the cells along the trajectory. The video on the right shows how the image files generated by one of the Stream modules can be downloaded and displayed in the notebook using just a couple of clicks. As the volume of publicly available single-cell data has increased in recent years, many researchers are focused on similar topics, such as immune cell and tumor interactions. There's growing interest in being able to harmonize or align different data sets that were collected in different labs or slightly different experimental conditions, but that still in theory describe similar biology or are expected to contain similar populations of cells. CONOS from the lab of Peter Karchenko is one such algorithm that uses a clustering and graph-based approach to link populations of similar cells from different experiments. We will make available a notebook that encompasses this workflow, utilizing GenePattern modules to perform the heavy lifting in the cloud while enabling users to interact with the results and configure and generate plots in real time. We've recently released notebook project functionality. A project is a separate environment that allows you to install packages and libraries and upload support files that will only be accessible within that project. This allows you to work with notebooks that may have conflicting dependencies without their interfering with one another. When you first log in, all of your notebooks and files are in a single default project, and you can create additional ones as needed. You can share projects with collaborators, allowing them to edit the notebooks and files within a project. And you can also publish a project, making it available for other users of the notebook workspace to find, copy into their own accounts, and adapt to their requirements. The GenePattern Notebook workspace has a wide variety of pre-installed packages and extensions, including scikit, numpy, pandas, plotly, seaborn, and others. If there's a package or extension you need that isn't provided, you can easily install it into your own notebook account. There are also kernels available for several Python and R versions. If you do want to install GenePattern Notebook locally as an extension to your own Jupyter system, you can either grab our Docker image, which contains a complete Jupyter plus GenePattern installation, or you can install using the pip or conda package managers. Two notes here, we also have a Docker image for the notebook repository, so people can host their own GenePattern notebook repositories. And also, you don't need to have your own GenePattern server in order to run the GenePattern Jupyter extension. It will connect any GenePattern server on the internet. For those of you who are computational biologists, all of the functionality I described to run and access GenePattern analyses is available through our Python library, and the code for all components of the GenePattern ecosystem is open source and available on GitHub. We continue to add new capabilities, analysis tools, and featured notebooks. Some new upcoming features include the following. The Jupyter Lab interface is the next generation of the Jupyter system. A beta version of the GenePattern notebook environment running on the Jupyter Lab interface is available at lab.genepattern.org. It uses all of the new Jupyter Lab functionality to organize analysis tools and provide a framework for future capabilities such as analysis job results, revision history, and real-time collaboration. We're also collaborating with the Globus team to add Globus-based file transfers within the notebook environment, which will allow robust transfer of data sets of any size from the many Globus endpoints around the world to a user's notebook account. In another collaboration, we're working with the Galaxy developers to allow users to connect to any Galaxy server and view and embed Galaxy tools within a notebook. This will make the thousands of Galaxy tools available as notebook cells, with seamless data flow between Galaxy, GenePattern, and Jupyter Lab. We are also incorporating the JavaScript IGV and Cytoscape visualization tools into the notebook format, making them available as cells within a notebook that can be seamlessly incorporated into a notebook or popped out for a more standard visualization experience. I'd like to acknowledge the GenePattern notebook developers and computational biologists in the Mesrov lab and all of our research collaborators. Thank you all for listening.